then you end up with those electrons being sent down the electron transport chain. And so when you think about an electron transport chain, you have to have like an ultimate source of where are those electrons coming from, so water, and then where are they going to end up? There has to be an endpoint at some point. And so in this case, it's set up so that NADP plus is the endpoint. That's the final electron acceptor, which works out really well because then if NADP plus accepts those electrons, it gets reduced to NADPH, which is also very important for the plant so that they can do part 2 of photosynthesis. You can see it's complicated. There's a lot of players in the game I always kind of think of it as like a hot potato game where they're passing the electrons from player to player in the game until finally NADP plus gets it and is very happy because that's the goal, to get your NADPH made. So these photosystems, photosystem 1 and photosystem 2, are both getting hit with light energy, as you can see on that diagram. And hopefully they're getting hit with pretty much the same amount of energy so that everything's going along really smoothly in terms of the electron flow from water to NADP plus. Okay. As this is happening, as the electrons are passing down the chain, the other thing that's happening as electrons are getting stripped off of water is that you end up building up quite a bit of protons as another product. You know, you get oxygen too. We're not going to think about that. But let's think about the other product, the protons. And so here, if you can imagine this happening over and over and over again, you would get a buildup of protons in the lumen. So much so that you have more protons in the lumen compared to what you find in the stroma. You also see that there's some funky things going on here with protons in the stroma, and they're showing that they get funneled or pumped across the membrane and end up in the lumen. So not only are there protons building up because of water getting stripped of electrons, you also have protons moving across the membrane from stroma to lumen. So you get protons disappearing from the stroma, you get protons building up in the lumen, and that's your proton gradient now across this membrane, all right? So anytime you start talking about a proton gradient across a membrane, whether it be the mitochondria membrane, jeez, this won't let me write. It's supposed to say proton gradient. Anytime you get a proton gradient across a membrane, here's the chloroplast, thylakoid membrane, that's potential energy available to do something. And it can do different things for the organism, depending on the situation. But in this particular situation, they want to use this potential energy to create ATP. So because protons are positively charged, they can't just diffuse across the membrane. So once you get this gradient, by the laws of chemistry, they want to diffuse back. They want to even out their protons across the membrane. But they can't do that going through just the regular portion of the membrane because they're charged. It's not compatible with that huge hydrophobic portion. And so they have to go through a transport protein of some sort. And in this case, it's the ATP synthase, which is a huge multi-subunit protein that spans the whole width of the thylakoid membrane. And as those protons are flowing through the ATP synthase, it actually creates a torque force. These subunits up here, they rotate and they allow for inorganic phosphate and ADP to come together and react. And if that happens, then you get that lovely ATP. So as the electrons are getting transported down the chain, this is also occurring hand in hand. Anytime you see electron transport chain process, look for a proton gradient. It's happening. Okay, you can't divorce the two. It's kind of a universal thing that organisms have evolved. And whether you be a plant or an animal, we all do it. And so it's really nice because not only are they using the electron transport chain to generate NADPH, by the process of that electron transport chain running over and over and over, you get a lovely proton gradient, which then allows for production of ATP. What do you think? Does that make sense? So if you've taken any other, phys classes, whether it's human or animal, plant phys, we cover this in a lot of detail. This is not as much detail as what we do in that class, but still, you have to think about kind of the big picture idea here of the plant has set up this situation with this electron transport chain process, and there's two goals it has in mind, NADPH and ATP. And as you can see, it's kind of complicated, but they got it to happen. Okay, so now the situation is set where if they have enough of that NADPH and ATP, they can start kicking into the biochemical reactions. Really, to be more specific, I think carbon fixation is a better way to describe it, because carbon fixation means you're taking in CO2 and fixing it or changing it into a sugar. Okay, so carbon fixation. So now this is going to be happening in the stroma portion of the chloroplast, right where NADPH is being made, ATP is being made, right in the stroma. So it's good that the carbon fixation is also taking place right there. And so that's very easy then for ATP to be used and NADPH to be used from the light reactions. And they're using it to ultimately, when you look at why are they doing this, ultimately it's so that they can get sugars. And so now that's the Calvin cycle that we're talking about. This is actually named after Dr. Melvin Calvin. That's his name. Poor kid. I bet he got picked on a lot on the playground, but he rose to great heights, became a biochemistry PhD, won the Nobel Prize for his work on this, and he actually did his bachelor's degree at Michigan Tech University. So he is no stranger to the UP. So Dr. Melvin Calvin was doing some work on the idea of how are plants making their sugars. And he joined in on a team of researchers that had made some progress on it, but they were stumped. They kept looking for this idea of, what could the CO2 be reacting with in order to get the sugars forming? And they were picking apart this physiological pathway, and they knew that the first stable product were three carbon sugars. So they're like, okay, we know the product, the initial step is a 3-carbon molecule. So we know that CO2 has a carbon, so we have to hunt for a 2-carbon molecule. So they hunted and hunted, looked for a 2-carbon molecule that CO2 could react with, and did a lot of work. Dr. Calvin said, well, let's back up a little bit. What if we think about a different way to get to the number 3? Maybe we could jump up to 6 and then divide it by 2, and that would give you 3. And so they started looking for, oh, maybe it's a 5-carbon sugar. And if you add CO2 to it, then you get 6 carbons. And then if you divide that by 2, you get 3. So they started looking for that. And the reason they missed it is that back in the day, they didn't have fine enough technology to pick up that there was an unstable molecule being formed, and it quickly broke down into 2 3-carbon identical molecules. So they missed it initially. And finally, the technology advanced. And so they did discover this molecule, ribulose 1,5-bisphosphate, thanks to Dr. Calvin's help on that. And so you can abbreviate that down to RUBP. Okay, so this is a 5-carbon sugar. It has 2 phosphates on it. So bi or bis means 2. and 1 and 5 means that's where the phosphates are bonded. So ribulose 1,5-bisphosphate, RUBP. So that's what CO2 is reacting with. So CO2 and RUBP react, and ultimately you're going to get some sugars. And in order to get all this to happen, you need to have ATP and NADPH. So this is the actual Calvin cycle. Sometimes they call it the C3 cycle because the first stable molecule is 3 carbons. So here, if we start at the upper portion, you see CO2 is coming in. Here's your RUBP. The gray are the carbons, so it's allowing you to count carbons here. And you can see that there's this unstable 6-carbon molecule that quickly breaks down into 2 identical phosphoglyceric acid or phosphoglycerate molecules. Okay, so you do need to know RUBP and CO2 gives you PGA. All right, you don't need to know about the short-lived one. I'm not asking you to really think about that too much. But I do want you to know that the CO2 is reacting with RUBP and that the first product there will be the 2 molecules of PGA. They're 3 carbons each. So the way the artist drew this to go from RUBP to PGA, do you see any ATP or NADPH being used? No. So that means that if there's enough RUBP and CO2 near each other, if they're hanging out with each other, they're going to react and form that PGA. They don't need any kind of input of energy of any kind. It's just going to spontaneously happen, which is good. The enzyme that facilitates this reaction is Rubisco. You have to know that, okay? And Rubisco and RUBP are 2 different things, okay? RUBP is your 5-carbon sugar. That's what CO2 is going to bond with. Rubisco is the enzyme. So I imagine Rubisco sitting there. It's going to take CO2 into its active site, RUBP into its active site, react those molecules, and then you get your PGA. Okay, so you definitely have to know that first step and the enzyme. So phosphoglycerate, another way to say that is phosphoglyceric acid. Anytime you see ATE in a science word, that usually means it's an acid. So that's why they labeled it PGA. It's the acronym for phosphoglyceric acid. Okay, so that's kind of phase 1 of the Calvin cycle. Phase 2 is to take your PGA and through reactions, get you your G3P. Okay. Now here you can see ATP and NADPH are needed. So to go from PGA to G3P, you definitely need ATP. And what NADPH is doing is donating some electrons. So NADPH is happy to come along and say, oh, you need a little electrons here to push this reaction. And ATP is there as well to phosphorylate. So by the time that phase 2 is done, you're going to go from PGA to glyceraldehyde 3-phosphate, which is abbreviated G3P. So please keep these separate. In the past, I've seen students switch those. They confuse PGA and G3P. So my advice to you would be to write this or type it over and over until you get it down in your head, the order, so that you don't mix these up. Okay. Now, this is a cycle. So that means we got to go back to the start, which I'm only showing you part of what's happening here, part of the goal. So part of the goal of the Calvin cycle is to cycle back to the beginning, which is to take some of that G3P and run it so that you can get more RUBP. Okay, so that's phase 3 of the Calvin cycle. And so the Calvin cycle has kind of a two goals to achieve. It needs to make more RUBP so that all that incoming CO2 all day long has something to react with. It needs to react with RUBP. So all day long, the plant has to keep this cycle going. So making more RUBP. And you can see here, this phase 3 portion, you can see several arrows here. So there's a lot happening there, but we're not going to get into huge detail in this class. Okay. So basically, by the end of that third portion of the Calvin cycle, you need to convert G3P to RUBP. So that's one goal of the Calvin cycle. The other goal, I'm not even really showing it on here. Let me go to this slide. Here's the other goal, which is this little offshoot. The other goal is to take some of that G3P, some of this G3P, and you can kind of see in this green box, they say after 3 turns of the cycle, 1 G3P is available for use in making sugars. That's the second goal of the Calvin cycle. All right. So the first goal is I got to keep making RUBP so I can keep this going. And then when they get enough G3P building up, they're going to take some of that and send it into another reaction pathway to make sucrose ultimately. Okay, so that's the other part, which I'm not going to get into the reaction pathways on that because it's making sucrose. That's what you need to know. We don't need to get into the physiology of that. Okay, questions so far on the Calvin cycle? Yes.
What's the point of the NADPH in the beginning for the light reaction gaining a hydrogen when it's just going to give it up there? Why doesn't it have a
So
just direct the hydrogen straight to this?
I know, right? But it's kind of what evolved millions of years ago. I mean, we do that too. We're doing that too in our mitochondria in the sense of well, it's more NADH that we make in our bodies. Plants prefer to make NADPH. But yeah, it's a nice mobile molecule that can carry electrons and help out in reactions nearby by donating off electrons into that reaction. So yeah, why not make it more simple and direct? It's just the evolutionary process that came up with this idea, so to speak. Perhaps a biochemist could answer that more clearly for you. But I mean, when you look at the light reactions too, it's like, why make it so complicated, right? But I did hear a biochemist say, and it kind of stuck in my head, and it makes sense. If you just rip electrons off of water and hand them all to NADP plus, and you look at the thermodynamics of that, the cell would like burst into flames. It'd like blow up because it's too much energy being released all at once. And so this is kind of a controlled process, which to me, it's like, oh, okay, that makes sense that they have to kind of control it and keep, kind of a little bit of energy until you finally run it to its completion. So yeah, but it's over millions of years that you had these reaction pathways building up and evolving and getting more complicated. Okay, so that enzyme Rubisco is actually, they hypothesized it's pretty certain about it, the most abundant protein in the world. When you think about all the organisms that do photosynthesis, not just plants, there's a bunch of cyanobacteria that use Rubisco as well. And so if you think about all the organisms that use Rubisco for photosynthesis, getting their sugars made, it's phenomenal. And just in the leaves themselves, they think about 40% of the protein that you find in leaves is Rubisco. So very, very important enzyme, which is another story of evolution because It's a weird enzyme, which we'll get into in a minute here. So and yet it's one of the most critical enzymes on the face of the earth. Let me back up. Okay, so this slide here, this picture, is the same thing that we just looked at where we were counting carbons and following the whole thing. It's just that they kind of broke it down into 3 phases. And so if this helps you for studying in terms of what's happening during each phase. So the carboxylation phase is just saying that's when CO2 is coming in and it's carboxylating the RUBP. So RUBP and CO2 are reacting and you have then the PGA. And then to go from PGA to G3P, that's what they call a reduction, primarily because NADPH is being used to give off electrons. So they are saying that's a reduction step. So, and then the last phase is regeneration. What they mean by that is you got to keep regenerating RUBP. So you can see that when you look at the cycle, the Calvin cycle, that there are 2 spots, 2 phases where you have to use products from the light reactions. It's here in the reduction phase where you use ATP and then NADPH, and then also when you have to generate more RUBP, you have to push it up with ATP. You have to use some phosphates from ATP to make more RUBP. So this is all happening in the stroma, the watery portion of the chloroplast. We have this cooking along all day long, hopefully. Right as the sun strikes the plant body, they'll open their stomata and allow CO2 to come in, and then this kicks off right away and goes all day long. And as this is happening, you get a nice buildup of the G3P, and then some of that can get sent into a different pathway to make ultimately sucrose. Questions on the Calvin cycle? Okay, so Rubisco is a funny enzyme. So photosynthetic carbon oxidation cycle. So sometimes Rubisco, most plant physiologists would say Rubisco makes a whoops, makes a mistake, and you'll get the kick in of this cycle, the photosynthetic carbon oxidation cycle. So when you look at the word Rubisco, I didn't have that anywhere. When you look at Rubisco, that's an acronym. So the RUB is, or Rubis, that's referring to the RUBP. That's, reacting with the CO2. But then the C in Rubisco stands for carboxylase. And then the O in Rubisco stands for oxygenase. So it ends in those 2 words end in an ASE, which means, this is enzyme activity. So this Rubisco enzyme, what it's saying by its acronym when they named it this, it's that they understood that this enzyme can work with CO2 in its active site, or it can work with oxygen in its active site. Either one, Rubisco's not picky. It's like, yeah, whatever. I'll take CO2 into my active site and use that to react with RUBP. Great. Oxygen comes in. It's like, yeah, okay, I'll use oxygen and I'll react that with RUBP. So from the plant's perspective, that's not the greatest. At least most plant physiologists would say that's not the greatest. And so this idea, what the biochemists would say is that carbon dioxide and oxygen are competing for the active site in Rubisco. Whatever Rubisco sees more of, whatever it's surrounded by more of, that's what's going to win and come into the active site. So if there's a lot of CO2 around Rubisco, great. It'll take in CO2 and react it with RUBP, and you got the Calvin cycle kicking off. If oxygen comes in to the active site and reacts with RUBP, you got a problem. And we call it the PCO cycle. And I'll show you why it's a problem. And so that oxygenase aspect of Rubisco. It's like, yeah, I can take oxygen too. I don't really care. It's all the same to me. And so whatever one gets in to the active site, that's what it will use to react with RUBP. So if oxygen gets in there into the active site, then it's going to react with RUBP. And you do get one molecule of PGA, which is good because that's the Calvin cycle. That's what you want. But then you get, instead of two identical molecules of PGA, you only get one. And then you get this funky two-carbon molecule called phosphoglycolate. And phosphoglycolate can't feed into the Calvin cycle. There's no enzymes to facilitate that. And so phosphoglycolate has to go into more of the PCO cycle. So the PGA that's getting produced here, that's fine. That's good because it can get fed right into the Calvin cycle. But that two-carbon phosphoglycolate will have to feed into the PCO cycle. Now we're going to see when you look at the PCO cycle, you do get some benefit from it. You get what they call a partial recovery of some carbon.
Is that oxygen just left over from the photo?
Yeah, it can be from that. Sure, from the light reactions. Especially since it's getting produced right there in the, the chloroplast. So they say there's partial recovery of carbon. I'll explain what that means. And then they say some carbon is lost. So I'll explain more when we are looking at the visual of it. So some people call the PCO cycle photorespiration. And so they're kind of using the analogy when we respire, like right now we're respiring. What are you giving off when you exhale? What gas? Carbon dioxide. So when they say some carbon is lost, they actually see the plant loses some carbon dioxide. Instead of fixing it into sugars, it's actually lost. And I'll show you what I mean by that with some visuals here. So that's why they named it photorespiration. So this PCO cycle is not a good thing. They've done many experiments where they have plants, identical plants, and they force one of the plants to run the PCO cycle. They know how to do that. And so they'll make that plant run the PCO cycle and they have the control, same identical plant living a more normal life where it does the Kelvin cycle mostly. And you can see a big difference by the end of, especially if you draw that experiment out for the growth period of the plant. So let's say 3 months. You can see that the plant that was forced to do the PCO cycle did not grow as well, far less biomass. And if it's an angiosperm, far less fruit produced in flowers, of course, and fruit produced. So it's considered a negative thing. It impacts the ability of the plant to finish its life cycle in a nice, healthy, vigorous way. So here's that Rubisco enzyme. And they're showing the active site. And they are saying RUBP comes in, which is great. And if you go the top route and CO2 comes in as well, that's great because that's what you want for the Calvin cycle. And you end up with two identical molecules of PGA. But if Rubisco sees a bunch of oxygen instead, then you end up with only one PGA and then a 2-phosphoglycolate. Not so good. Here's another graphic trying to get that point across. And you can count your carbons on this one, right? So here's your two molecules, PGA, and here's only one. And then you got that funky 2-phosphoglycolate. And if you like to do it this way, if you're more of a chemist, where you want to see it this way, this is another way to show it. And then if you prefer a flow chart, here's another way. So I'm devoting several slides to this concept. This is a very important concept, all right, because it really makes a huge difference to the plant on whether CO2 is coming into the active site of Rubisco or oxygen. And so you can see here, if there's High levels of oxygen, not much carbon dioxide. You end up with that phosphoglycolate, and you can see arrow, arrow, arrow. There's a whole lot of stuff happening in this section right here. So this arrow, 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 where they say that phosphoglycolate has to continue on into the PCO cycle. Those of you who've had plant fizz, this should look familiar. That's the PCO cycle. It's actually, two ways to look at it. This is a more pared down version over here, but it's saying the same thing. And no, you don't have to memorize all this. Yay! But this is the PCO cycle in totality in two different ways of looking at it here. Okay. So what we just looked at in that previous slide, let's go back to this one here. So Rubisco and oxygen, and you get one molecule of PGA and then that phosphoglycolate. Let's go back here. So then here's RUBP and oxygen reacting. And you can see one of the products is PGA, which is great because that can feed right into the Calvin cycle. But we only get one PGA. The other product is phosphoglycolate. And you can see that phosphoglycolate, the arrow, arrow, arrow on that other side, that's all this. Has to go through, with the use of 3 organelles, all of these different reactions until finally it builds enough carbons and rearranges things well enough that you get another phosphoglycerate forming. But in the process, you lose some carbon. I wish the artist had been a little bit more exaggerated about the CO2 here coming off. That's a big deal right there. So this is the lost carbon they're talking about. The plant respires the CO2 instead of using that to fix it into sugars in the Calvin cycle. It's lost. And so they see a reduction in how well the Calvin cycle is running and how much it can build up its sugars. So when you force a plant to do this, it suffers. It doesn't grow as well, biomass is lower, yield is lower. It probably is more susceptible to issues like drought or cold or pathogens when it's struggling. So in general, you don't want a plant running this all the time. Have I convinced you? Okay, we're done. So thank you for your patience, and we'll continue this on Wednesday, and then Wednesday we'll also do some review for the exam in case you have some last minute questions.